All right, hello everyone and welcome to our March session. So just as a reminder, the March Myco talks are coming up on the 4th working Thursday of the month. I'll put the information into the chat, but those are great things to sign up for and they always have a fantastic lineup of speakers. So we highly recommend those. With that, I will turn it over to Liz, who's going to introduce our first speaker.
Great, thanks so much, Jess. It's my pleasure today to introduce Shannon, who is going to be talking today about Candida-induced changes in bone marrow compartment, directing trained innate immunity against sepsis. Shannon's currently a postdoc with Marin Pekmezovic at the Tulane University. Before that, she did her PhD with Andy Alspa at Duke University and on Cryptococcus. So hopefully we'll be in for a really fascinating talk. Take it away, Shannon.
Okay, great. Let me just share my screen really quickly. Okay, and you guys can see my PowerPoint slides?
Like you're good to go.
Perfect. Okay. All right. Well, thank you so much, Liz, for that introduction. And I just want to take a second to thank you guys, the organizers, not only for giving me this opportunity, but also just for putting on the seminar series every month. I've really enjoyed it and I'm really excited to be a part of it now. And so as Liz said, my name is Shannon and I'm a postdoc in Marin Pekmezovic's lab at Tulane University. And today I'm going to tell you about my work looking at how Candida is inducing changes in the bone marrow to direct trained innate immunity against sepsis. And so in the Pekmezovic's lab, we study mechanisms of immunomodulation and inflammation caused by Candida species during the host pathogen interaction. And as this audience is well aware, Candida species can cause a variety of infections ranging from mucosal infections of the oral or vaginal cavities to systemic or invasive candidiasis. And in our lab, we think a lot about the concept of the damage response framework, which defines microbial pathogenesis as a function of both the microbe and the host, where microbe-mediated damage results more often from a weak host response, whereas host-mediated damage can result from too strong of a host response. And so these different types of Candida infections can be represented by different damage response curves, where they can result in varying levels of primarily microbe-mediated damage, primarily host-mediated damage, or a combination of the two. And while over the years, our lab has studied many of these different types of candidiasis and their associated curves, we've recently focused a lot of our attention on invasive candidiasis of intra-abdominal origin. And so intra-abdominal candidiasis is triggered by Candida invasion of the abdomen from the GI tract, which can be caused by surgical procedures or insertion of medical devices such as catheters. Outcomes range from localized peritonitis to disseminated candidiasis. And important for this talk, and unlike invasive candidiasis of hematogenous origin, intra-abdominal infections are often polymicrobial, and in particular, bacterial co-infections are very common. And when fungi are involved in these infections, mortality is significantly increased. And in addition, fungi contribute to worse outcomes, longer hospital stays, and increased antimicrobial use. And so if left untreated, these infections can lead to fungal sepsis, which is characterized by host-mediated damage and organ dysfunction resulting from a hyperimmune response, including high levels of inflammatory cytokines. And so this fungal sepsis represents the right side of this intra-abdominal damage response curve. And our recent work has been focused on how to reduce this host-mediated damage. And so to study this, several years ago, our lab developed a mouse model of polymicrobial intra-abdominal infection with Candida albicans and Staphylococcus aureus, in which when we inoculate mice intraperitoneally with either Candida or Staph alone, they are generally fine and survive the infection. But when we inoculate them with Candida and Staph together, the mice die very quickly. And this is due to a robust inflammatory response and high levels of pro-inflammatory cytokines, both shown here locally, but also systemically. And so this is indicative of a lethal sepsis. And so in further studies, We began investigating whether this synergistic lethality was shared with other non-albicans Candida species, which led us to discover that the closely related Candida dubliniensis actually did not induce the same level of synergistic lethality with staph. And in fact, we went on to show that if we inoculated mice with C-dub prior to lethal co-infection, we could actually protect them against lethal sepsis. And so just to briefly summarize some of the work that our lab has done over the years to define the parameters and limitations of this protection, we found that this protection is not specific to Candida albicans, but that we see protection against polymicrobial intra-abdominal infections with other synergistically lethal Candida species. We find that protection requires live C-dub. We've tested a number of Candida species in both our intra-abdominal model and in protection, and we find that protection is shared to some extent with other low virulence Candida species, including various Candida albicans mutants and Candida goprata. We find that protection lasts up to 60 days, but starts to wane after 90 days, and that it's maintained following multiple lethal polymicrobial infections. And we can also protect against bloodstream candidiasis using the IV infection model, but we don't see good protection against vaginal candidiasis. And while all of the work I'm going to show you today was using the intraperitoneal route, we have shown that if we administer C-dub by IV, we can actually also provide protection. And so as for what's mediating this protection, we found that it's actually not dependent on adaptive immunity, shown here with C-dub inoculated RAG1 knockout mice that maintain survival through lethal co-infection. We've also found that protection is not dependent on macrophages, as demonstrated here, where we clodronate depleted them. However, when we antibody deplete GR1 positive leukocytes, we lost protection, highlighting a role for these cells. And so the most common cells that express GR1 are neutrophils and myeloid-derived suppressor cells, or MDSCs. And considering that neutrophils tend to have a very short lifespan, we've focused a lot of our attention on these MDSCs. And so if you're like me and you don't necessarily consider yourself an immunologist, you've probably never really heard of MDSCs. So I'm just going to take a few slides to describe these cells to you. So these are a heterogeneous population of immature myeloid cells that share the common biological activity of suppressing immune responses. They are characterized by the co-expression of GR1 and mature immune cells, which makes them difficult. And while they're most well known for their role in cancer, they have also been well established as having a role in sepsis, sometimes in reducing sepsis. And important for this talk, they've also been shown to accumulate during fungal infections. So MDSCs are thought to develop through a two-step process. First, the population of immature myeloid cells in the bone marrow is expanded, in part due to a block in the further differentiation towards mature immune cells. The second step is activation, in which the expanded immature population becomes pathologically activated and their suppressive functions are expressed. And this is thought to occur in the periphery, so at the site of inflammation or infection. In our case, this would be in the peritoneal cavity. And so we hypothesized that C-dub is serving as the signal for this initial expansion step, and then lethal co-infection with Candida albicans and Staph aureus, pathologically activate these cells. And importantly, in addition to being a step in the development of MDSCs, this expansion of the myeloid compartment and changes in myelopoiesis have also been demonstrated in several models of trained immunity, just a small sampling of which I've shown here. And so we decided to start by looking to the bone marrow to begin to understand how C-dub is inducing protection. And so as I mentioned, we inoculate our mice intraperitoneally with C-dub. And actually, on my very first day in the lab, we discovered that C-dub can actually traffic to the bone marrow. So we were able to culture it from bone marrow at 24 and 48 hours after intraperitoneal inoculation. And we further showed that C-dub can actually penetrate the bone marrow at a higher level than Candida albicans or Saccharomyces cerevisiae among some other non-albicans species we tested. And in fact, we found that the level of bone marrow penetration actually correlated with the percent protection provided by various Candida species in our intra-abdominal model. And so we next wanted to see what was actually happening inside the bone marrow compartment. And when we looked at the total hematopoietic stem and progenitor cells in the bone marrow, we found that the entire compartment was expanded following C-dub inoculation. And actually this occurred in a dose-dependent manner where the more C-dub you inoculate with, the greater the bone marrow expansion. And we further showed that protection is also dose-dependent and aligns nicely with this expansion data, where those C-dub inocula that induce the highest levels of expansion are the most protective, while those that do not induce an appreciable level of expansion are not protective. And so we next wanted to know what specific populations within the bone marrow were affected by C-dub being there. And so again, we inoculated mice and collected bone marrow over time, and then assessed the hematopoietic stem and progenitor cell populations in detail by flow cytometry. And this diagram is just showing the different hematopoietic stem and progenitor cell lineage populations, starting from the most primitive and pluripotent hematopoietic stem cells, and then down to the more committed progenitor cell populations, along with the different markers that I used to identify them. And I'm going to walk you through which of these populations I saw changes in. So differentiated and undivided population shown here in this purple box and often referred to as LKS cells based on their markers. I found that this total population again was over time after C-dub inoculation. Looking more closely at the multipotent progenitor subpopulations, which are technically still uncommitted, but they can have a bias towards a certain lineage, I found that the myeloid-biased MPP3 cells in blue were increased, and that this occurred at the expense of the lymphoid-biased MPP4 cells in green, indicating there's likely an increase in myeloid progenitors in the bone marrow. Going further into the more committed progenitor populations, I found that the common myeloid progenitors, or CMPs, shown in pink, were initially decreased, and that this occurred concurrently with an increase in the granulocyte macrophage progenitors, or GMPs, shown in blue, which suggests that the CMPs are being rapidly differentiated into the GMPs. And so altogether, we're seeing that C-dub is inducing an overall expansion in the bone marrow and a bias towards myelopoietic lineages. And so considering this expansion of the myeloid compartment, we also looked specifically at putative or immature MDSCs in the bone marrow, which you can identify based on CD11b GR1 staining. And so we also found that these cells were expanded following C-dub inoculation, occurring a little bit later, but maintained throughout the time course. And so now it's in my title, but I haven't really discussed the concept of trained innate immunity yet, so I wanted to take a minute to do that. But basically, the definition is a non-specific memory immunity that's mediated by innate cells that have been trained by an initial challenge, leading to an enhanced response to a secondary challenge. And so graphically, that looks like this, where there's an initial response to an initial stimulus followed by an enhanced response to a secondary stimulus. And so I next wanted to test whether these changes I was seeing in the bone marrow cell populations actually had any functional consequences. And so to do this, I again collected bone marrow cells from mice at different time points after C-dub inoculation. And then I challenged them in vitro with live Candida albicans for 24 hours. And then I looked at a panel of cytokines in a multiplex ELISA assay. And actually, I found that many of the cytokines in our panel were not changed, with the exception of the 4 that I've highlighted here in blue and orange. And so among those 4, I saw an interesting pattern where the anti-inflammatory cytokines in orange tended to continually increase over time, whereas the pro-inflammatory cytokines in blue more tended to peak early and then trended back down. And so I think that this data is showing us a few things. First, that the bone marrow does have an enhanced secondary response, that's indicative of trained innate immunity. And second, I think that this initial pro-inflammatory response is actually in response to C-dub being present in the bone marrow, while the continually increasing anti-inflammatory response is really what we're interested in terms of how C-dub is providing protection. And so I think that the relevant enhanced response is primarily this anti-inflammatory response. And to that end, of note, we also found that this anti-inflammatory response in the bone marrow seems to be specific to C-dub. So when we collect bone marrow from mice inoculated with Candida albicans or Candida tropicalis and re-stimulate it in vitro, in this case with Zymosan, we actually find that the increase in anti-inflammatory IL-10 is only found in the C-dub bone marrow. And unlike the C-dub bone marrow, which has reduced pro-inflammatory TNF-alpha, Candida albicans bone marrow actually has enhanced TNF-alpha. And so this suggests that this anti-inflammatory response is C-dub specific. And this is interesting and important because not only is this different than Candida albicans, but it's also different than other previously published trained innate immunity inducers, such as beta-glucan or the tuberculosis BCG vaccine, which tend to be more pro-inflammatory. And so we next wanted to confirm whether this bone marrow expansion was actually required for C-dub mediated protection. And so I used an antibody against c-Kit to deplete the hematopoietic stem and progenitor cells. And I administered this antibody at time points after C-dub inoculation. And I was able to show that it could block expansion in the bone marrow. And so I next challenged these mice with a lethal co-infection, and I found that C-dub mediated protection was in fact abrogated, indicating that C-dub induced bone marrow expansion is at least in part required for protection. So far I've shown you that C-dub can traffic to the bone marrow where it induces an expansion of hematopoietic stem and progenitor cells, a bias towards myelopoiesis, an expansion of immature MDSCs, and an enhanced anti-inflammatory response. And so bringing back to this graphical representation of trained innate immunity, we actually think that C-dub is mediating a slightly different flavor of trained immunity, which we've taken to calling trained tolerogenic immunity, where the secondary response is more anti-inflammatory. So unlike traditional trained immunity, which tends to be more pro-inflammatory, thereby reducing microbe-mediated damage on the left side of this damage response curve, trained tolerogenic immunity acts to reduce host-mediated damage on the right side of the curve. And so we think that MDSC precursors in the bone marrow are expanded and trained by this initial C-dub stimulus. And then mature MDSCs are mediating the trained tolerogenic response following lethal sepsis challenge. And so I haven't really talked much about these mature MDSCs, and that's partially because I didn't want to go over in time. But what I will tell you is that we have found that both peritoneal cells in general, and specifically GR1 positive putative MDSCs, are increased in the C-dub mice following sepsis challenge. And so really the big open question is, are these functional MDSCs? And so you guys will have to invite me back to share all of the data one day, but we have begun characterizing some of the known functional markers of MDSCs. And we've started to be able to check off some of these boxes. Most importantly, for the MDSC field, we've been able to show that these GR1 positive cells can suppress T cell proliferation. And so we're really excited to finalize and start sharing that aspect of this project as well. And so in summary, what I've shown you is that following IP inoculation of C-dub, this fungus traffics to the bone marrow where it induces training and expansion of hematopoietic stem and progenitor cells, a bias towards myelopoiesis, an expansion of immature MDSCs, and a primarily anti-inflammatory response. And then following IP inoculation of Candida albicans and Staph aureus, these putative GR1 positive MDSCs traffic from the bone marrow to the peritoneal cavity, where they limit inflammation and suppress immune responses in sepsis. And so there are still really a lot of open questions. First, we're really trying to understand the mechanism of hematopoietic stem and progenitor cell training in the bone marrow, including trying to identify which cell types specifically and which receptors are involved in the C-dub interaction. And then as a major hallmark of trained immunity, we're also pursuing studies to assess whether there are epigenetic changes induced in the hematopoietic stem and progenitor cells. And then on the MDSC side, we're still trying to further functionally characterize these cells. So looking at additional functional markers of suppression. And I didn't really get have time to get into it here, but there are different subsets of MDSCs. And so we're also trying to characterize which subset is involved in our intra-abdominal model. And so with that, I would like to thank the Novair and Fidel Labs, and then also all of you for your attention. So thank you.
Thanks so much for that fascinating talk, Shannon. I'm sure there'll be lots of questions to ask after the second half of our talk. I'll pass it back to Jess for now, who's going to introduce our next speaker. If you have any questions to ask Shannon, please go ahead and put them in the chat, and we'll read them out just after Marin is done speaking.
Thank you, Liz, and thanks, Shannon, for a great talk. I'm very pleased to introduce Marin Pekmezovic today. So Marina earned her BSc and MSc in molecular biology and physiology at the Faculty of Biology, University of Belgrade. She then gains exposure and learned about human fungal diseases through her work as a research associate at the National Reference Library for Medical Mycology. She also served as a treasurer for the Serbian Society of Medical Mycology. And she moved to Jena, Germany for her doctoral work at Friedrich Schiller University. After completing her PhD, Marina then joined the HKE to study invasive fungal infections. And so today, Marina is going to tell us about her work on vaginal candida. Marina?
Thank you very much for the introduction. So I have not much to add, apart from that I'm also very, very honored to be here, and I like this seminar series a lot, and I'm very happy to show my data. So the majority of data, or basically everything that I'm going to show, was obtained during my PhD at the MPM department. And at the very end, I'm going to introduce what I'm doing now as a postdoc in a junior research group, Adaptive Pathogenicity Strategies. So first, I would like to drag your attention to epithelial cells, which are going to be the main actors of today's stories. So epithelial cells, as you can imagine, are in a constant interaction with different microbes that can be beneficial, strict pathogen or opportunistic pathogen or commensals. And they are definitely more than simple physical barrier. They are actually an immunologically responsive barrier that is responsible to, as a key players, to sense and modulate the innate immune system. And then when we look even more specifically to the vaginal microenvironment, it is highly dynamic. It has a lot of fluctuations, not only during the lifetime of a woman, but also on the daily or weekly basis. And this environment is very complex also due to different pH changes, oxygen availability, hormonal influence, conditions such as pregnancy or menstrual cycle. And therefore, when these conditions are disturbed, some of the pathogens, especially the opportunistic pathogens, can cause the infection. And of course, today the topic is vulvovaginal candidiasis, and there is no need to introduce much. You know that this infection is characterized with very high morbidity and a high recurrence rate. It's not lethal, however, it significantly reduces quality of life. And based on epidemiological data, it is mostly caused by Candida albicans that I also have here at the video, and you very well know all the main virulent traits of this pathogen. However, reports show that Candida glabrata is the second most common, followed by other species such as Parapsilosis tropicalis and others. So what is actually happening? So in the commensal state, there is a very low fungal load, and therefore there is no immune response initiation. However, during the infection, Candida is overgrowing, producing hyphae, which is causing epithelial damage, and this is recruiting the cells of innate immunity. And therefore, the whole environment is shifting towards pro-inflammatory. And what is also specific for vulvovaginal candidiasis is that there is a very large infiltration of neutrophils that are recruited. However, they are unable to actually solve the infection, and therefore the VVC is actually considered to be immunopathology that results from a very aggressive innate response by neutrophils. And all this actually suggested us that epithelial cells, as being as a gate between the pathogen and the innate system, have a very important role, and we wanted to investigate this in more detail. So for this purpose, I used an in vitro model. So this model is already very well established and already studied. So basically, it's a monolayer of vaginal cells that can be infected. And in this case, depending on the research question at the different time points, interaction between Candida and vaginal cells can be studied. And as I said already previously, this model provided a lot of valuable information about adhesion, invasion, and damage caused by Candida albicans, and actually made us to understand more. However, there were some gaps, and with my PhD project, I tried to answer them or to fill them. So first, on the fungal side, majority studies, or basically all of the studies, were focused on Candida albicans only, excluding the other species. And therefore, one of the main questions was, what is happening then with other Candida species? How are they behaving? And on the host side, in the similar direction, the scenario that I showed you, again, of course, is true for Candida albicans, because this fungus is producing hyphae, Candida lysine, and so on. However, other fungi, such as Candida glabrata, they are not producing hyphae and not producing toxin, but they are still causing symptomatic infection. So the question would be, what are then the mechanism of the host response to different Candida infections? And my project was had aimed to include other Candida species, to also couple this to transcriptional profiling in order to get more information about it. And this will be the part one of my talk. And then in the second part, I will explain how we actually wanted to find out a little bit more about the fungal side and fungal virulence by increasing the complexity of our model. So speaking about the comparative transcriptomics, so we basically did dual RNA-Seg. We isolated RNA from the fungus and from the host at different time points. I used 4 different Candida species that are mentioned here, and for the host, vaginal epithelial cells. And first, I'm going to explain the less exciting part, which is a fungal response. So here you can see the processes that were upregulated in each fungus at each time point. And as you can already see at a glance, that there is not really much overlap. There are only some of the processes that I labeled here with the arrows that were in common for at least 3 out of 4 at some point of infection. However, the majority of the processes seem to be species specific. And this was not really a surprise. We followed in detail some of the clues that we got, but we, of course, confirmed basically things that were already existing in the literature, and therefore concluded that each species had its own way how to behave during this interaction. And this further supports the idea of that evolution of pathogenicity of each of these species actually was independent. So more interesting data we got on the host response. And for the purpose of this talk, I'm going to focus at the early time point, 3 hours, because I would not have time to go into all details. And this was definitely the most interesting. So showing you the same graph, but now speaking about the host response. So here you can see that at the early time point of infection, host cells seem to respond pretty similar to all 4 species. And this was very interesting, and we had a closer look. And we saw that the majority of those processes were actually related to mitochondrial processes and type 1 interferon response. And this was very interesting because actually those 2 terms come together when speaking about the initiation of the immune response. So for those who are not familiar, mitochondria are playing a very important role in the immune response. So they can basically sense many different stressors, including infection. And in this case, mitochondria are releasing mitochondrial DNA. And this mitochondrial DNA can then trigger different signaling cascades, including a Seg-sting axis, which results in the initiation of interferon response. And initiation of interferon response basically means the upregulation of interferon-stimulated genes. And those genes are coding for the proteins that are involved in a variety of different processes. And apart from antimicrobial immunity, also cancer, inflammation, autoimmunity, and so on. So we definitely observed an upregulation of those genes, but we then wondered, okay, let's have a look what is happening to mitochondria. So first, just by staining the mitochondrial network, we could already see that there was definitely a difference between uninfected cells and infected. You can see that the mitochondrial network is more fragmented and localized around nucleus. Also on the transmission electron microscopy, we could see that compared to the healthy mitochondria, mitochondria in infected cells seem to be more rounded, and they also had loss of integrity. This already indicated that they might be transiently dysfunctional. So we also performed some additional tests. For example, measuring mitochondrial membrane potential, we saw that it was decreased. So there was depolarization. We also saw that there was an increased mitochondrial ROS production and significantly increased release of mitochondrial DNA into the cytosol. So this it's very important to mention that this change was transient and it was selective. So it was not that all mitochondria in the cell did that. So it did not lead to apoptosis. And this gave us a sign that obviously mitochondria are releasing this DNA in order to spread a specific signal. And then we wondered, how do we connect this with the upregulation of IgGs that we are seeing? So we tried to find a proof that mitochondrial DNA actually is a connector between these two processes that we observe. So we transfected vaginal epithelial cells with the mitochondrial DNA, and we could see that there was a very strong upregulation of IgGs, and it was much stronger than if we would use nuclear DNA. And on the other hand, we also treated vaginal epithelial cells with DDC, and DDC is causing depletion of mitochondrial DNA. And when we would infect these kind of cells that have depleted mitochondrial DNA, we could see that there was no upregulation of IgGs. And this all proved us that mitochondrial DNA indeed is actually connected and needed for the initiation of type 1 interferon response. So putting this all on the scheme, our data indicate that Candida is somehow recognized by vaginal epithelial cells. We are not sure which kind of receptor and how the signal is induced. However, it causes this transient mitochondrial dysfunction, which results in release of mitochondrial DNA that is later on initiating type 1 interferon response, most likely via Seg-STING axis, but we did not test this in our model. And this all results in the secretion of interferon beta, which is then binding to its receptor IFNAR, and this causes signaling cascade, which results in the transcription of interferon-stimulated genes. And this happens autocrine, so on the same cell, but also on the neighbor cells, therefore spreading the signal. So having all this in mind, we wondered then what would be the role of this pathway during the interaction, why it's initiated, and what can we do with this finding. So we thought, okay, what happens if we block it? So we used experiments with blocking antibodies, so blocking IFNA receptor, or silencing some of the IgGs that were the most highly upregulated. And in this case, we could see that this was detrimental. The epithelial damage was increased if this pathway was blocked. On the other hand, if we would pre-treat epithelial cells with interferon beta, and therefore inducing the IgG expression before we infect, then if those cells would be infected, the epithelial cells, the epithelial damage was reduced. So it indicated that this pathway can be protective. And this was actually very nice in accordance with a published study a few years ago, where in a rat model of vaginal candidiasis, they used interferon alpha, which is also type 1 interferon, and it was shown to be beneficial for the treatment of VVC in rats. So to summarize this first part, I showed you that the fungal response was highly species specific with not so much overlap between the species. However, on the host side, we saw a uniform response at the early stage of infection, which was characterized by selective mitochondrial dysfunction, which led to initiation of type 1 interferon response, which was protective. And then we thought we were not very satisfied that on the fungal side, we did not really have enough data. So of course, we worked at the same time on another project, and we thought to focus on a non-mainstream Candida species. So we thought, what about glabrata? As you can see from the epidemiological data, it's the second most common. Of course, the percentages are varying depending on the study, but this was actually very interesting because Candida albicans, we all know it produces toxin, it has high Phe, it's invading, it has very strong, causes very strong immune response. So it has, let's say, all the right to be the most common one. But Candida glabrata is everything opposite. So has no hyphae, no toxin, it's evading immune system, it's actually causing very weak damage, or basically in our model, almost no damage. It was also shown almost avirulent in mouse models. So we then wondered, how is it possible that it's the second most common? That it might be that our models miss something, and this could be maybe some specific human factors. So thinking about the environment in general, again, in the context of vaginal mucosa, apart from epithelial cells and candida cells, we also have cervicovaginal fluid. And this is a body fluid that is very rich in many metabolites and proteins, such as immunoglobulins, mucins, antimicrobial peptides, and albumin. And albumin was particularly interesting because it's present in all the body fluids, and also there where Candida is interacting with the host. And it was already shown before that it can modulate growth and virulence of different pathogens, including Candida. So we thought we will include it in our model and see what happens. And it was very interesting to see that only by addition of albumin, the damage caused by Candida glabrata was significantly higher, and it was reaching the level of Candida albicans damage. And this was true for the bovine's albumin as well, but not for the murine. At the same time, we observed an increase in adhesion and increase in growth. But what was interesting, that Candida glabrata could only grow better with albumin if the host cells were also present, otherwise not. So to make a very long story short, I'm just going to summarize on this scheme what we found. And our data showed that epithelial cells are taking up albumin, they are processing it, and they are releasing oligopeptides and amino acids. And this is then used by Candida glabrata, which is increasing growth and causing epithelial damage. Then we performed also RNA-Seg, and this gave us hints that also genes involved in oxidative stress resistance, virulence, adhesion, and some metabolic processes were also significantly upregulated. And furthermore, we also tested deletion mutant library. So we tested more than 600 glabrata deletion strains in this model. And this led to the conclusion that HAP5 was actually the key protein mediating this augmented damage by increasing iron availability during this process. So this data actually contributed with to, let's say, finding out another model of damage which was independent of hyphal production and toxin secretion. So to sum up my work, I aim to dissect the interaction of vaginal epithelial cells with 4 most prevalent Candida species. And I showed you that they had fungal specific, species-specific pathogenicity mechanisms, while the host response was very similar at the early stage. At the later stage, this was not the case, but I did not speak about this today. If someone is interested, we could discuss. But additionally, I also showed you that addition of a single factor can turn non-virulent species into a virulent and increase the damage, which was mediated by iron availability and proteolytic degradation. And finally comes the question, what is the next step? So obviously type 1 interferons caught our attention a lot. And as you could also hear on the previous seminar series, and in general, it's becoming also a very hot topic in our field in general. This graph here shows the role of this pathway in different bacterial infection. And you can see that the picture is definitely not black and white. It's very, very complex. And also if we think about in vivo environment and interaction and coexistence of Candida with different viruses, this again points out that this pathway for sure is very important to modulate antimicrobial response in general. So it would be very interesting to follow up on this pathway. And therefore, what I'm doing now for my postdoc, I'm trying actually to see if those interferons could have immunotherapeutic potential. And I'm focused on intestinal colonization, intestinal model. And actually, Shannon had a very great introduction for me. I don't have to say more details. You know that this could be a step that leads to systemic infection. Therefore, the project is focused on investigating the translocation of Candida albicans through the gut barrier. And in this case, I want to investigate not only type 1 interferon, but also type 2 and 3, how they can modulate this, and if any of them could have a therapeutic potential. And with this, I'm at the end of my presentation, and I want to thank Professor Huber, who supervised my PhD project and provided me with the best conditions I could have. Also to Mark, who also partially supervised my PhD project and now supervises and is the leader of the group where I'm a postdoc. Also to Celine and Sasha, included also in my supervision. My master student Anne-Christine, that performed a lot of albumin experiments. To Opathy Consortium, where my PhD was part of, and among them, especially to our collaborators in Barcelona and Dublin, and to all the people from the MPM and APS department, to funding, and to all of you for your attention. Thank you very much.
Great, thank you, Marina. That's a really fascinating talk. I like the interplay between albicans and glabrata. So I'm going to pass it back to Liz for questions for Shannon. And if everyone would put their questions into the chat, we'll go back and forth between the two speakers. Thank you.
All right, Shannon, we've got a bunch of questions here. I'm just going to read them off as they come in. First question is from Jessica. She says, fascinating and frightening data. Do patients with Candida albicans infection show an increased risk of hematopoietic cancers if they survive their initial infection?
So that's actually a very interesting question that I've never thought about before. And I don't really know the answer. And I think that's partially because I'm not sure that people are really looking at this. There's not really a lot of data about Candida in the bone marrow in general. So I think that's a really interesting question that we should maybe look into.
Yeah, maybe there's some epidemiological data there in terms of patients who've had anti-Candida drugs that that are more enriched for particular kinds of cancers later on in life.
Yeah.
I'm afraid my connection wasn't very good, and I just wanted to clarify a concept that you discussed early on in the talk. Do you know exactly how the cells are getting into the bone marrow again? Is that clear about how it's being trafficked?
So I didn't put that in my talk, but a lot of people have asked us that question, and we have done some experiments where we have depleted macrophages and PMNs, and CDUB still gets to the bone marrow. So we actually don't know how it's getting there, but it doesn't seem like it's like a Trojan horse mechanism.
Okay, that's really interesting. I've got one more question, then I'll pass it to Jessica. So Mark Swiggerdahl has asked, he says, beautiful presentation. Did you test if IL-10 knockdown mice or other anti-inflammatory knockout mice lose the protection? So is trained immunity related to beta-glucan? And are these differences or branching in beta-glucan from CW, are there differences in beta-glucan branching from C. abrogates and other fungi, which are less protective?
Okay, so it's actually very interesting that you ask if we tested IL-10 knockout mice, because I actually had that in my talk and then I took it out because I didn't know if I had enough time. And I will tell you that we have tested IL-10 knockout mice, and IL-10 is required for protection. So we do lose the CDUB mediated protection in these knockout mice, which we're really excited about. As for the differences in beta-glucan between CDUB and albicans, I'm not very familiar with how different the CDUB cell wall is from Candida albicans, but it's something that I've actually thought a lot about, and I think could be really interesting to look into. I will say that we have done some experiments with beta-glucan in our lab where we have modified the beta-glucan, and we see different responses, different levels of protection, depending on whether we have modified or unmodified beta-glucan. And I think this could actually maybe be key to the differences that we're seeing. Oh, and yeah.
Yeah, so the last part of that is, will IP inoculation of C. abrogates beta-glucan induce MDSC expansion and protection? So can you just give this as a therapy, basically?
Yeah, that's like my dream experiment, actually.
All right, let's give Marina a chance. We'll come back to this with the rest of the questions.
All right, thank you. So Marina, first question from Paul Fidel. He asks, what is the fate of the immunopathology during infection if the epithelial cells are protected by the interferon beta treatment?
Okay, this is a very nice question. So we did not really follow up much on how this protection, or let's say the epithelial cells treated with interferon beta, would modulate the immune response. What we did, I did not show this due to limitation in time, but for example, if we would have epithelial cells that had anti-IFNAR antibody, so we would block the whole pathway, and we take those supernatants and we put them on neutrophils. For example, islate production was significantly higher, which can indicate that maybe this pathway, the whole type 1 interferon pathway, has some kind of an anti-inflammatory role. In this case, maybe it can modulate commensalism. This would be one of our hypotheses. However, we did not really follow up further on how this would influence the recruitment of neutrophils or whatever, but this is definitely our plan to further see and maybe really have the interferon beta potentially as a regulator of commensalism.
They're very exciting data. And then Mariano Affiari asks, do you know the mechanism of the protective effect of interferon beta? So you just answered that. Is there a reduced fungal burden following co-culture with epithelial cells and interferon beta?
Reduced fungal burden, I have to say I did not quantify it, but at least from microscopically what I see, I don't think that it was reduced. What could be the mechanism, and we are trying also to answer. So as I said, once this whole pathway is initiated, it results in the transcription of the genes that are ISGs, right? The interferon stimulated genes. And these genes have a variety of different roles. For majority, we have absolutely no idea what they do. But I would say that probably the answer is hidden there. So we plan to follow up on the top candidate one and to see if we could really follow up on mechanism. And maybe this can be related to differential mucus secretion. Maybe this could be the protection mediated, or any kind of change of the expression, what is on the surface to reduce the adhesion or something like this. So I would say that this is very, very exciting field, and probably one of these ISGs could give us this answer.
Mm-hmm. And the second part of Mariano's question was, are there reduced markers of apoptosis or necrosis in epithelial cells with, plus interferon beta following co-culture with fungi?
So I have to say we did not really measure the apoptosis. We only measured the apoptosis in general, and we did not really see it at anyway in the normal conditions, let's say without interferon beta. But it would be interesting to see if maybe necroptosis maybe, or any kind of special cell death mechanism would be then involved in the whole system. But yeah, this is a very good point, and this is definitely worth to follow up on.
All right, thank you. I'll send it back to Liz for some questions for Shannon.
All right, so we've got a few questions that are kind of in the same vein. James Masuka has asked a similar question to Mark, which is, would you expect to see a protective effect from CDOB cell wall extract? Although I'm not sure how you deliver that to the bone marrow. And I just to kind of put another twist on that, have you tried heat-killed CDOB, or is it required, is live cell required?
So again, using the cell wall extract, I think would be a super cool experiment, but I think how it would get to the bone marrow could be a problem. As for using heat-killed CDOB, we do know that CDOB has to be alive in order to get the protection.
Okay, really interesting. So Chain Shin has asked whether pre-inoculation with a low dose of Candida albicans can do the same thing as this high dose CDOB, or if it's a species-specific phenomenon.
So we have also done those experiments, and we do see some protection when you lower the dose of Candida albicans. The problem that we have is sometimes the Candida albicans kills the mice in the primary infection. And so that's sort of why we haven't, I mean, yeah, that's a problem.
Mm. Yeah, can I just clarify, are you curing these mice of the infection through like caspofungin or something, or is this just
the CDOB?
Yeah.
We're not curing them. The CDOB gets cleared because we do the secondary like co-infection 14 days later. And we've done a bunch of experiments, and we see that CDOB is cleared mostly like from different organs at different times, but it's pretty much cleared by 14 days.
Okay,
So it's
you're
not there anymore.
anticipating that it's in the bone marrow and growing at the time when you're doing this.
No, we actually, I think it's cleared from the bone marrow like within 7 days.
Okay. So just the final question in this round, Shannon, Matt Blango says, Shannon, really cool talk. Any concept of how widespread the phenomenon of trained telerogenic immunity might be? He has to assume it won't be unique to just fungi.
Yeah, so I'm not super sure about trained tolerogenic immunity, but I will say that just generalized trained immunity is not specific to fungi. It's been shown to be induced by beta-glucan, by the tuberculosis BCG vaccine, by some viruses. As for this more anti-inflammatory form, I also don't necessarily think it's just specific to fungi. I just think we haven't quite figured out what specifically is inducing it yet. Like what about C. diff is inducing the anti-inflammatory response? And I think once we figure that out, then I'm sure there are going to be other things that do the same thing.
Yeah. Okay, thanks so much. I'll just pass it back to Marina, and then we'll have one more question later on.
All right. Axel Dietschmann asks, might the AIM2 inflammasome instead or in addition to CGAS sting be involved in the signal transduction after mitochondrial DNA release?
Yes, this would this could be a possibility. I'm just not very sure, or at least all the inflammasome markers that I tried to test in the vaginal epithelial cells simply were not, or let's say I could not really find anything. For the CGAS thing, I said we did not really prove it in our model for the simple reason that it's regulated post-translationally. So we were mostly based on the transcriptional data. However, I did have staining with the phosphorylated antibodies against the antibodies against phosphorylase sting, and I could see the increased signal, but we did not really follow up on this. So I would say that probably goes via CGAS sting, but of course it could be possible that other mechanisms are also involved. I just think that maybe in the vaginal epithelial cells, it would be a little bit hard to show that.
Yeah. And then I was very curious about your HAP5 involvement, if I caught it correctly, since HAP5 is part of a complex that regulates a bunch of different respiratory genes. And so did you see similar mitochondrial involvement, or do you think that glabrata is responding metabolically differently than albicans? Or what are your thoughts on that pathway?
Yeah, of course, you're absolutely right. It's in the center of everything. So of course, we expected that once we use the HAP5 knockout, we would have significant decrease, for example, in growth or whatever, but we did not see. So for example, HAP5 mutant grew the same like the wild type, everything was same. The only difference was that once we add albumin, it did not have an increased damage. And it grew like less than the wild type in the sense of augmented growth. And we are now following up on this in the terms of oxidative stress resistance. Mm-hmm. We think that this is actually the key there, that maybe the lack of HAP5 is making them less resistant to oxidative stress. And we think that actually this increased resistance could be one of the mechanisms why they can actually support this increased proliferation in the presence of host cells and albumin and actually cause the damage. And if this is not very well regulated, they cannot support that, they cannot damage and further replicate. But we did not really check the mitochondrial look or genes or whatever, but we definitely know that the metabolism is different based on the transcriptional data and that oxidative stress resistance differs.
Very cool. All right, I think Liz will take the final question. And if you have more questions, either put them in the chat or turn on your mic, please.
Yes, Shannon, so the final question is from Kelly Shepardon. She says, thanks for a great and interesting talk. I was wondering if you did histology or looked to see if C. dub remained as a yeast in the bone marrow. And what's the answer to that?
So we have histology, but I'm not sure that we've looked very in depth at whether it's still a yeast in the bone marrow. That's a really interesting question. Kind of want to go look at all those slides now.
Yeah, because the second part of that question is if you used a yeast-locked albicans, would you see a similar protective effect? And maybe it's the hyphal form that's sort of preventing this trained immunity from doing what you want to do.
Yeah, so we have used yeast-locked strains of Candida albicans, and they are quite protective. We use them in some of our intravenous experiments.
Interesting.
We haven't looked actually to see if the bone marrow expansion part of this whole story is the same in the yeast lock strains. But because we do see protection in some of the other Candida species that also form hyphae, we don't think that it's just that it's yeast locked. I do think that it's maybe something to do with the cell wall changes that are happening in the yeast locked Candida albicans mutant versus in C-dub.
Interesting. Yeah, because of course the cell wall organization of yeast locked is going to be very different from hyphal
Yeah,
albicans, even within that species. It's a great insight. Okay, that's the last question. If anybody else has anything they want to add, I think you can just turn your camera on and fire away. And if not, then I think we'll just thank our speakers so much for two really fascinating, beautiful talks. And hope to see some of you online or in person next week at the Fungal Genetics Conference.